Hi everyone, welcome to Dento'ZEN. Today we are starting with a new subject, that is medical microbiology. What do you think? What is microbiology? Micro means very small. Biology refers to the study of living tissues. So yes, microbiology is the science which deals with the study of those small living organisms which are actually the causative agents of infectious diseases of humans. And when inside the humans, they can cause some reactions, and also we should know the methods of protection against such diseases. So we can say that microbiology deals with three things. First is the study of causative agents of infectious diseases, second is their reactions, and third is the methods of protection against such diseases. Before we discuss microbiology in detail in general, we must first know a little bit about the history of the microbiology as a science, how it was developed. So that we are going to do in today's video. So let's start. Before starting, quickly subscribe to Dento'ZEN, tap on the like button, hit the bell icon so that you remain notified about new videos. Now these organisms are so small. Do you think we can see them with naked eye, direct eye? No. Definitive knowledge about these bacteria became possible only with the development of microscope. But then, who was the first person to see these bacteria? Don't forget, it was Antony van Leeuwenhoek who first observed and reported bacteria. He had an interesting hobby of grinding lenses and seeing different materials through them. He also gave them a special name. He called them little animalcules. So those can be your important entrance and viva questions. Who first observed reported bacteria and what term was given? Now microbiology as a science was developed by another important person, that is Louis Pasteur. He evolved the basic principles and techniques of microbiology, introduced different techniques of sterilization. Sterilization is the method by which any area is made free of bacteria. So he developed steam sterilizer, hot air oven, autoclave, which are different methods of sterilization, out of which hot air oven and autoclave are two important long questions in microbiology, which we are going to discuss in detail in our next videos. He also invented pasteurization of milk and urine. Pasteurization is the technique by which it is partially sterilized to make it safe. He also coined the term vaccine and discovered anthrax vaccine and anti-rabies vaccine. So many contributions by Louis Pasteur. So application of Louis Pasteur's work was done by another person, that is Joseph Lister, who introduced the antiseptic technique in surgery. So that is another important contribution. He used carbolic acid for antiseptic technique and reduced the death rate. Now another important person in microbiology is Robert Koch. Why? He was the first person to introduce solid media, introduce staining techniques and methods of obtaining bacteria in pure cultures using solid media using agar, and he discovered bacteria like tuberculosis, bacillus, cholera, vibrio, and he was the first person to do hanging drop operation, a method to see the motility or the movement of the bacteria. So many contributions by Robert Koch. So he is known as the father of microbiology. That can be your important viva and entrance question. Who is the father of microbiology? Robert Koch. So attracted by the work of these two important persons, Louis Pasteur and Robert Koch, many other scientists discovered different bacteria. So let's get to know which other bacteria were discovered. Hansen described leprosy bacillus, then Neisser described gonococcus, Oxton discovered staphylococcus, Loeffler first isolated diphtheria bacillus, Nicoler first observed tetanus bacillus in soil, Frankel described pneumococcus, and Shordin and Hoffman discovered spirochete of syphilis. So with the discovery of so many bacteria, it became possible to lay down some criteria to associate specific bacteria with specific disease. So that was done by Koch. Koch gave postulates, criteria which are known as Koch postulates. Initially four criteria were given, later on one additional criteria was added. So what are these postulates? These are the criteria by which a microorganism can be accepted as a causative agent of an infectious disease only if that microorganism follows these five criteria, which can be your important viva and entrance question. What are the Koch postulates? So let's get to know. First point is that this bacterium should be constantly associated with the lesions of the disease. That is, this bacterium should be able to produce the same lesions of the disease every time constantly. Second is, when we try to take these bacteria from the lesions, we should be able to isolate it in the pure cultures. So it should be possible to isolate this bacterium in the pure culture from the lesion. So that is the second point. Third point is, from these pure cultures, if we try to inoculate these bacteria into the lab animals, it should be able to produce the same lesions of the disease. So that is the third point. The pure cultures into the lab animals should reproduce the same lesions. This disease should always be produced again in the animals. Fourth point is, from these lesions, if we again try to re-isolate these bacteria in the cultures, it should be possible to re-isolate these bacteria in the pure cultures from the lesions in the experimental animals. So those are the 4 points. Now, another point was added that a specific antibody to these bacteria should always be demonstrable in the serum of the patients, in the blood of the patient suffering from the disease. So, these bacteria should be able to produce specific antibody against them. So, these are the 5 points. First is it should be constantly associated with the lesions of the disease. Second is that it should be able to be isolated in the pure cultures. Third is it should be able to reproduce the lesions of the disease in the lab animals. Always disease should be reproduced in the lab animals. Fourth is it should be re-isolated in the pure cultures. And fifth is a specific antibody to these bacteria should be demonstrable in the serum. So those are important Koch postulates. So by the beginning of 20th century, many infectious diseases were proved to be caused by these bacteria, but certain diseases has still had unknown cause. So it was said that there could be some organism which is too small to be seen even under the light microscope. There came the term virus. So Bejering was the first person to coin the term virus, and yellow fever was the first human disease proved to have viral etiology. Then these viruses were not only causing infections, but they could also produce cancers. So Alleman and Bang were the first to put forth this concept that virus can cause malignancy. So definitive study of these viruses were made possible only with the introduction of electron microscope by Raska, because they were too small to be seen with light microscope. Now these viruses showed a phenomena, that is they caused the lysis, breakdown of the bacterial cultures. They attacked the bacteria. So this lytic phenomena in bacterial cultures was observed by Watt and D'Herelle. Another important phenomena, that is when the organisms are inside the human body, they are engulfed by the cells of the human body, that is called phagocytosis. This was discovered by Koch. Along with that, there became the discovery of different types of blood groups. So Karl Landsteiner is the person who gave the theory of blood groups and discovered rhesus factor. Those can be your important entrance and viva questions. So don't forget Karl Landsteiner with blood groups. Then parasites were also discovered. Another important discovery came from the Ronald Ross. He discovered that transmission of malarial parasite which causes malaria, its transmission can be done by the female Anopheles mosquito. Another important factor that was discovered in blood is called as humoral factor, which is also called the term antibody. That was done by von Behring and Catasto. So many bacteria were discovered, viruses were discovered, phenomena, reactions were discovered, blood groups were discovered, and then there was also the discovery of first antibiotic, that called penicillin. So first antibiotic, that is medication against this bacteria, was penicillin, which was discovered accidentally by Alexander Fleming. That can be your important viva and entrance question. So that is in brief about the history of the microbiology. Now let's check what have you learned. So which out of these is not a Koch postulate? We already know the Koch postulate. So first says that bacteria should be isolated from the diseased tissue. Yes, it should be. Second is when inoculated in the lab animals, bacteria may or may not produce disease. How is that possible? It should always be able to produce disease. Third is pure cultures can be obtained, and fourth is bacteria should be able to be isolated from diseased tissue in experimental animals. So first, true, it should be isolated in diseased tissue. Second, it should be able to produce disease. So no doubt should be there. Third is it should be obtained in pure cultures, and fourth is it should be isolated from the diseased tissue in experimental animals. So which one do you think is not a Koch postulate? Next question is, who is called as the father of microbiology? So the options are Robert Koch, Joseph Lister, John Hunter, and Alexander Fleming. So that is all for this video. If you really enjoyed the video, do tap on the like button and share this video with your friends. Subscribe to Dento'ZEN. Keep following, keep watching, keep learning. See you in the next video on microbiology soon. Till then, take care. Bye bye.